Okay, so our next speaker is Eva Kanso from the University of Southern California, and Eva will talk to us about spontaneous transitions in fish schools. Eva, the floor is yours.
Thank you so much. Thank you, everyone, and thank you for having me and for inviting me. I'm a little bit new to this community somehow. So I am going to tell you one project. I will not overload you too much. It's already the last day of the meeting, and it's going to be about fish schools. So these are the type of collective phenomena that I find fascinating, and I'm sure most of you do too. So if you look at this flock of birds, look at those beautiful density waves that propagate. The entire population kind of expands and contracts and does this beautiful collective behavior with, it's not clear what is the purpose of these and why we have these density waves back and forth. And we see similar phenomena in fish schools. We see some phenomena like this in herding sheep. If you notice, if you have very good eye, you might notice there are a couple of dogs running around here that are responsible for actually herding the entire group. And we see similar stuff at the cellular level. So I want to, biologically, people think that there is use for this type of collective social behavior in finding mates, evading predators, foraging for food. But I'm interested in the mechanisms that facilitate this type of behavior. So in the biology community, or the community that thinks about the interplay between mechanics and neuromechanics or neuroscience, we often see this type of loop, of feedback loop between information available to the organism, to an individual organism, and the behavior of that organism. And usually it goes from the organism is interacting with an environment, you have the biomechanics of the interaction with the environment, but also the organism is collecting information via sensory modalities, whether it is about its external environment or its internal environment. And this information is typically processed in a central nervous system that gives commands to a neuromuscular system to act on the environment. And you have this information behavior loop, and that underlying this loop, there is an action perception cycle or a perception action cycle. And of course, one important ingredient that we are hearing a lot about is the role of memory and learning in this process, and this is still a big challenge for us. So we have looked at different aspects of this centralized intelligence, if you like, in application to specific behavior, because it's very difficult to try to understand behavior as a whole. You narrow it down to behavior that's repeatable and that's really testable in a lab. So we have looked at feeding, at predation. We have looked at evasion strategies. I don't know how much you can see my notes here. We have looked at locomotion, at navigation in unsteady environments, especially flow environments. But today, I really want to focus on schooling and tell you a little bit about the work on schooling. So this is a shift now from thinking about individual intelligence to thinking about collective intelligence, or centralized versus decentralized intelligence. And I will just to repeat myself, this is a talk about emergent collective behavior in fish schools. And before I go ahead of myself and talk too much, I want to acknowledge my lab, my students. The student that did most of the work that I'm going to present today is right here, Chen Chen. This is an old picture from when he started his PhD. And the other topics that we look at in my lab are at the interplay between physics, sensing, control, and understanding the mapping from structure to function in biological organisms, and at different length scales. We're interested at large length scales, like fish and birds, but we're also interested at a very small length scale. And I hope that this is a story I get to tell you some other time. So let's start with our model. We're doing modeling. We're doing basically mathematical models of this that try to combine the different ingredients, the physics part, the sensory part, and the control or the response in collective fish behavior. And this is work that I started with Christophe Eloi, or Christophe Eloi started with me a couple of years ago. And this is an animation that you are going to look at where we have about 1,000 swimmers. Each one of them is creating a flow field. So it has a flow, it's creating a dipolar flow around it, and it's going to interact with the flow that's generated by all other swimmers. And this is the one example of behavior that you could obtain. You start from an isotropic initial condition. There is no structure in the initial condition, and then the school organizes. And I forgot to mention, it's not only flow interaction, they are also responding to visual cues about where the others, to their immediate neighbors. So they see their immediate neighbors and they respond to visual cues, and I'll talk about this in a second. So this is one type of collective phenomena that you could see. You see there's a large structure that's emerging, and you have those, the density is concentrated towards the edge of this milling behavior. There are many interesting phenomena that if we look closely, we see happening in this type of emergent behavior. But what we could tell by doing a more thorough parametric analysis on this model is that we actually obtain this 3 typical collective behavior that people have observed in different models. We're not the first to do this. People have looked at the emergent collective behavior in self-propelled particles from the Vicek model and before the Reynolds model, all the way to Ian Cousins' work and lots of other recent work. And people have observed a swarming behavior where the entire school basically stays together, but everyone is in their own, doing their own thing. Maybe it's like what's happening here. I don't know if you're with me or in your own world, but that's the swarming behavior. You have some cohesion, but everyone is pointed in a different direction. Schooling is when the entire group is organized and polarized to move in one direction. Milling is some of the behavior that I showed you now. It's where the entire school goes around. And we have a new phase that we observed in this work that we did with Christophe, which is a turning phase, that the entire school actually turns, where you have both rotational order and polarization order, which was not possible before we introduced the hydrodynamic interactions. So we think that this new phase is really due to hydrodynamic interactions. So if you are curious about what's in the model, what have I been showing you? We have a simple particle model where every individual has a position r, index I for individual I. It's moving its own self-propelled speed, and it's also being advected by the flow created by everyone else in the school. And it also, the individual has a heading direction, theta I, and this heading direction is changing, and it's changing according to 3 effects. One is that the individual has some free will, if you like, in changing its orientation randomly, and that's modeled as a noise in this equation. And then you have the ability to orient with what your neighbors are doing. And here, by neighbors, I mean only the immediate Voronoi neighbors, your closest neighbors, basically. You want to orient with them or align with them. And then there is a third term here that's coming from flow interactions, because the flow field creates gradients, and if you are an elongated body and a gradient flow, that will tend to change your heading and orientation. And if you work a little bit, so one thing about this visual field, other than that it's really in response to immediate neighbors, it's also anisotropic in that you see neighbors ahead better than you see neighbors behind you. So there is this anisotropy in the visual field. And if you work a little bit hard on this set of equations, you can use some non-dimensional length and time scales that are not necessarily the physical length scale of the fish, and you can reduce the equations to mostly two parameters. Actually, you have a parameter about the intensity of the dipolar field that you're creating, but we're keeping that fixed. So we have now two parameters. One tells us about the alignment intensity, which is your propensity to actually work with the group and conform with your friends and with your neighbors, and your noise intensity, which is a measure of how much you have free will and you don't want to respond to what your neighbors are doing. So on one extreme, you can be completely independent and not conform with the group. On the other extreme, if you have no noise, you'll be doing exactly what your neighbors, exactly what the rules in response to your neighbors. So if some of you are actually fluid dynamicists and are worried about me modeling the individual fish as a dipole and telling me, why is that a good model of a fish? Because a fish, when it's swimming, it's generating a lot of swirling of the flow field and other stuff. Is it truly a dipole? So I have this image to show you. This is the fish in the red contour here. This is from the work of Ulrike Müller. She was one of the first people to actually measure the flow field in PIV, using PIV in actual experiments. And you see that at the head of the fish, you have a dipolar field it's like a bow wave, biologists call it. You push the fluid ahead. And at the tail, there is a lot of stuff that we are ignoring. And we are acknowledging that we are ignoring this stuff because this is not easy to keep track of. So as a first order model, we're taking the far field flow due to this movement of the entire fish body. And we are ignoring any wake that could be left behind. Okay, so this is the model. These are the results that we have already had a few years back. But when we started working with biologists, and here we are working with Matt McHenry at UC Irvine, this is the type of data that he would be able to present to us, where he's looking at the behavior in a lab experiment and a circular tank, and you see the presence of the boundary, and you see that these fish like to actually follow the tank wall, which is not one of the behavior that we see in unbounded domain. This is another experiment from Ian Cousins' group, where you see an entire school that's also, you have now a square boundary, and they kind of mill a little bit, they swim a little bit in a polarized way, and then they interact with the boundary, they change their behavior. And in this paper, there's this interesting switching between polarized schooling and milling. And what they reported is that the fish actually, this transition between the two states is dependent on the size of the school. So in a smaller school, you have a lot more transitions between the two states. In a larger school, that you don't have as many transitions. So this got us thinking about, well, what is the role of the environments, of the boundaries? Can you use boundaries to design new collective behaviors for the entire population? So this is what I'm going to focus on today. I want to understand how confinement and boundaries affect the collective phases that people talk about in fish schools. So I'm going to use the same model that I just presented to you, but I'm going to add circular confinements. I am honestly modeling the no-slip boundary condition, the slip boundary condition, but at the circular domain, so the fluid cannot go across the boundary. I'm also modeling the reaction of the individuals to the boundary. So those are the two new ingredients in the model. And you see that we get the four characteristic behaviors that we see in unbounded domain. No surprise. I'm showing you the streamlines here just to show you that we're honestly solving this. And the entire school has a flow signature, if you like. One thing that we noted even previously is that when you have this hydrodynamic interaction, the entire school is going to swim faster than when they are, on average, faster than when they swim individually. This may be relevant. But what we also saw in confinement, that we have these new phases that emerge that we don't see when we don't have confinement. One of them is following the tank wall, like we saw in the experiments of Matt, but we also see this double milling, which I find super interesting. The school splits into two populations. One population goes in one direction, the other population goes in the opposite direction. And it's not like a 50-50 split, it has some randomness, but this is like kind of a anti-phase synchronization and double pendula, if you like. But here it's happening in an immersion, in a collective system. And we also see, when we showed this behavior to Clémencire, he said, oh, you must be seeing an elongated milling phase. So we went back and we actually looked very carefully to make sure that this is not the elongated milling phase, that they actually do observe in unbounded domains, and it's really topologically different. They are not the same. You have an elongated milling where the fish just do this type of go around, they turn, and then they come back and turn again, kind of like a knotted trajectory. So we can characterize these emergent phenomena using polarization order parameters. One is the polar order parameter, how much they align together, the rotational order parameter. I showed them previously from Ian Cousin's work without introducing them. So we have a mathematical definition for these parameters, whether they are built on experimental data or model like this. We can also look at the locations that the school visits in its domain, and we see that yes, when you are schooling, you're visiting more of the entire domain. When you are milling, you are confined, visiting only a small part of your domain. So if you don't want to make yourself known by moving too far, you just do this milling motion. And those order parameters are very good at distinguishing these four different behaviors. They can really pick up swarming from schooling, from turning, from milling, because in one case, in milling, for example, you have high rotational order, low polarization. In swarming, both are low. In schooling, you have high polarization, but low rotational order. And when you're turning, both of them are high or intermediate. So to really characterize the three new collective phases that we saw, we had to introduce a couple of other order parameters, which we call them the confinement order parameter and the directional rotational order parameter, to be able to distinguish between these different phases and really characterize them uniquely based on their signature, basically. Okay, so that's done, but what's most interesting in this work was this beautiful surprise about a school that's going in this milling phase. This is, we're choosing one value of those parameters, the noise and alignment parameter. And this school is going to mill for a while. The order parameter here is very high, but now it's going to transition spontaneously. I'm not doing anything. I'm just doing forward time integration. And it's going to transition spontaneously to this polarized state. So the polarization order parameter is going to become high. And then it's going to transition again. If we wait a little bit, it's transitioning, it's going to transition again to this milling state. So this is the intermittent or spontaneous transition that I really wanted to show you today, that this happens immediately, spontaneously in the fish school. And this is a very different flavor of bistability. So this is saying that both the milling state is stable, but also the schooling state is stable, and you're transitioning between the two of them. This is a very different flavor of bistability than if you were to start two different initial conditions and you go to one state and stay there for the rest of the simulation time, or you start from a different initial condition and you go to another state and stay there for the rest of the simulation time. This we call bistable. Both are bistable, but just for notational convenience, I'm going to call this intermittent bistability, and this is just simple bistability. Okay? So this has a dynamic signature, the difference between these two. It has a dynamic signature in the intermittent bistability. When you look at the polarization and rotational order parameter, they are intermittently going from, kind of like we saw in the experiments, going from high polarization to low polarization, and then from high rotational order to low rotational order, back and forth. Now, in the bistability, where you don't see this switch, if you look at one realization or one initial condition, you don't see changes in those order parameters over the simulation time. Maybe a change might happen later, but over the simulation time, you don't see that. So we wanted, before we go more deeply into understanding this bistability, we wanted to understand what's the effect of confinement on actually creating these global phases. So what we did with Christoph here, with the no confinement case, we looked at the entire parameter space that we defined, which has the noise parameter and the alignment parameter. At every point in this parameter space, we're doing Monte Carlo simulations where we have 100 different initial conditions. We take the time trajectories of these 100 different initial conditions with 100 fish in each case, and we look probabilistically at the behavior that we obtain, and we label whether it's milling, schooling, turning, or swarming. And we reproduce exactly the same parameter space that we had in a previous study with no confinement. Now, when we add confinement, when we add the circular domain, but we have no fluid, and we can do that because we're doing mathematical simulations or mathematical models, you can knock off whatever term you want, you see that this turning phase disappears, which is consistent with what we had found with Christoph before. But you see the emergence of this following the tank wall phase, and you see the emergence of those bistable and intermittent phases. So these are boundary-induced effects. So for the same value of your parameters, for the same individual fish behavior, you can get bistable or intermittent, depending on where you are. And if you add the fluid plus confinement, you get a much richer, you get back this turning phase that we see in the presence of fluid. Now, one surprise was if we change now the number of fish, you have new phases that emerge. You have this elongated milling and double milling. Now you could ask me, I mean, isn't there like a density, like a number of fish per area that could govern the behavior? And why would we want to look at just numbers? So yes, we looked at that. We looked at whether what is the effect of density. So actually we wanted to see if density is the control parameter here. So the contour lines, the black lines that you see here, are constant density, and I'm plotting this on the space of the confinement radius and the school size. And you see if you have no change in behavior along a density line, doesn't prove anything, because that's one specific set of parameter values. But if you do have a change in behavior, then you know that density is not the correct control parameter, like in the VCheck classic model. And indeed, what we see here is that we have situations where we are along the same density contour, but we see a change in behavior from milling to elongated milling, or from milling to double milling, while keeping the same density value. So it means density alone is not your control parameter. You really have to worry about the domain size, and you have to worry about the number of fish in that domain. So that was a little bit surprising. So now, coming back to my intermittent, my bistable states, intermittent versus just simple bistable behavior, we wanted to try to understand like how do we characterize these different behaviors. And one way, what we were doing to show you those characterizations, we were basically computing probability density functions of the values of the rotational order parameter and the polar order parameter that we get over all our Monte Carlo simulations for a given parameter values, set of parameter values. And what we see is that these joint probability distribution functions, they cannot distinguish between intermittent and no intermittency. And this is not because of lack of numerical accuracy or not integrating long enough. This is really a fundamental limitation of using histograms or probability density functions, because in this case, you don't see the dynamics, whether you can switch. It's because you cannot see this behavior. So they both look the same. You get the same probability density function. So we wanted a way to actually characterize the dynamics and not just to look at those histograms. And of course, one way is to look at building a Fokker-Planck equation, kind of coarse graining of the behavior onto the space of polarization, the rotational polar order parameter and the translational polarization. So on that space, that's kind of like a coarse-grained space for the entire population. And we can think of each state, milling or schooling, as two different states on that space. And we want basically an equation that governs the transition between these two states. And the Fokker-Planck equation, what it tells us, it's the probability of being in one state at a given time. And the state here, my state, big X, the vector here, is this plane, basically, the two coarse-grained order parameters. Now, the difficulty is in deriving the drift vector and the diffusion matrix from first principles, from my microscopic equations. They are too complicated. Luckily, we have now, we're in the machine learning and the data age, we have methods to compute the drift and the diffusion matrix numerically from the data that we collected from our simulations. So we did that. And it turned out that the problem is even simpler than looking at a Fokker-Planck on a 2D space, because when we look at these PDFs, probability density functions on this space, we see that really the data or those variables are confined to a 1D subspace. So we can do a nonlinear projection onto a one-dimensional subspace of this 2D space. And we can write now a Fokker-Planck equation in terms of this reduced polar order parameter K, that when at one end it could be 0, it's milling, and at the opposite end it indicates schooling. And to understand the switching between these two states, we wanted to build a potential field to understand how the stability of these states. So this potential field, we're going to build it from our diffusion matrix and velocity drift vector. And you see that I'm coming back to the same figures over and over just to hit the point that when you have this intermittent case and you plot a probability density function over this projected order parameter, you see that it's double peaked, the same as when you have a bistable case. You see it's also double peaked, but that does not tell you about the possibility to transition between the two or not. But if you compute the effective potential of the Fokker-Planck equation, you see that in one case you have a finite potential barrier between the two states, so you can basically hop over this potential barrier, and in one case you have an infinite potential barrier and you cannot hop over these two cases. So you're in one state or in the other. So now we can use these tools to build what kind of like a way of changing each parameter in the model at a time and looking at the behavior, the coarse-grained behavior at the school level. So how does the behavior change as we change the noise intensity? How does the group level behavior change as we change the school size? In one case, we see that as we increase the noise intensity, we go from a state where we have two stable equilibria, like global equilibria, if you want to call them that, and we go from this bistable state to an intermittent state, and then we collapse through some bifurcation into only a schooling state. And this is what I'm showing here, where I'm taking 3 different values from up here, and you see that I'm showing the 2D space with the 1D projection, and I'm showing the potential function over this 1D space. You see the infinite potential barrier, finite potential barrier, and then a bifurcation where you go to only schooling. So larger school, larger noise, which is surprising. More noise, you pump more noise into your system, you have fewer collective states. Yeah? I thought it was surprising. You put more noise and you reduce the number of collective states. You reduce the ability to transition between those two different states. Now, if you increase the group size, you also go from bistable to purely milling, exactly like I showed you in the experiments of Ian Cousin, where they were showing empirically that you do get, as you increase the school size, you get this transition from bistable, going back and forth between milling and schooling to only milling. All right, so what's next? So this is the story I wanted to tell you. This is the work that was just accepted, and we're very happy about it. So what's next? So now we can play all sorts of stuff with these types of models. We can ask about propagation of information within the school, let's say in response to a predator that is, let's say in response to a predator that's now moving. I think I'm confusing my computer with two. So here we go. We have like a milling state. We bring in a predator, very naive predator. It doesn't respond to the school, but the school responds to the predator in a very typical way. So this is just a preliminary work on this front. We also got interested, just for fun, why not, to look at what happens when you put these schools in different geometries that are not simple circular geometries. Let's say you have like an obstruction between in a bi-chamber domain, or you have one chamber here, and the ability to transition between them is a very small, narrow obstruction. And I know that some people are interested in this type of problem to look at residence time in each chamber. So we can see this type of behavior. Sorry, let me see if this is going to play. I don't know why this is not playing. All right. Okay, and you see that you have this transition from one side to the other, and then you get another transition and back and forth. And those transitions, surprisingly, they are not due to vision, because these obstructions, they kind of prevent the individuals from seeing who's on the other side of the wall. So these transitions are mostly facilitated by flow interactions, because when we take the model and we turn off flow interactions, we see fewer transitions between the two sides. So this is just in the making. I don't have a complete story to tell you that. But we are also interested in taking these models to extreme scales in schooling. So this is an example, a simulation with 20,000 individuals. This is all done not on superclusters. We're all doing them on our own machines in the lab, so we're very proud of our efficient algorithms. They are multi-core machines, but this is still n-squared type simulation where you have interaction between individuals, where just through clever manipulation of the terms, we can reduce it to being computationally manageable to an n log n type simulation. And you see this type of behavior. Let me, before actually I run it, I wanted to say there is nothing different between the individuals in each part of this domain. The reason we put color is only to see if there is mixing, if there is interaction between these individuals that start initially at very different ends of the domain. And this is what you see. So we run it forward. This is for the same parameter values that I showed you with the 1,000 school that went on milling. This is 20,000, same parameter values. And now you see that we don't get this coherent milling behavior. The school actually expands, and then it starts to come back together. You have this mixing between individuals. You have what looks like rarefication density waves, where the density is rare, and then you have those kind of what looks like density shock waves. I don't know what's the sound, what's the speed of sound in a medium like this, but they look like density shock waves, and we're starting to look into that. Thank you. So at some point, I wanted to tell you about a bouncing star, and then I am going to not tell you about it. Just I want to maybe say here that this is in one organism that has many, many, many legs. They are called tube feet. And they cooperate together on pushing the organism kind of in the same way that many, many members of a school cooperate together going in one place. So this is really the same as this collective transport in ants. At least it is possible for the individuals, the individual tube feet, because this organism has no central brain. Sea stars and the echinoderms, they have no central brain. They have just a neural net. So it's very difficult for them to process all the information from all their feet. So the feet act almost as individual autonomous systems, and kind of similar to what we see in human behavior. I think we are thought to be the only species that cooperate to do transport, humans and ants. So we see that in the sea star. again, I'm going to thank you for listening to me, and I want to stop here and take any questions and let you enjoy this simulation of a 10,000 fish schooling together.
Okay, so thank you very much, Eva, for a fascinating talk. Are there any questions? Patrick?
Yeah, just to clear something up in my mind. When you were plotting those effectively one-dimensional probability distributions,
Yes.
was it a log scale or a linear scale? Have a motivation for us.
It was not a log scale for the PDF. It was not a log scale.
The motivation
Yes.
let you go explore those probability landscape, even if the probability wells are and the barrier between them is very large.
Mm.
There are methods for
I would be very interested in learning. Yes. Yes. I would very much appreciate those references. Yes. Thank you. Can you please repeat? Yeah, there is no inertia. This is a kinematic model. Yes, it will, of course, make a difference. Yes, it will make a difference. Yes. That's a good question. Not in my simulations, because I have a large attraction term. So you are attracted to your Voronoi neighbors. So if you're very, very far, you're going to still have your Voronoi neighbor. You're going to have a larger incentive to come back. And that we see with the larger schools that actually they split, but then they come back together, kind of like what we see in actual fish.
Swimmers that would never use that program with very rapidly. Then they wouldn't need to know where they were. That's really hard to do underwater.
Of course. Yes. Yes. There are lots of people that are doing all sorts of underwater application. It's still, this work is supported by the ONR, the Office of Naval Research in the United States. And now they are interested in having multiple vehicles working together. So it's still in the research state. We still cannot have thousands and tens of thousands of robotic vehicles going underwater and working together. Maybe we will have 2 or 3 in the next 10 years. But the Navy is putting a lot of effort and funding to actually get those to work together and to understand if there is benefit for the flow interaction. What information? It's kind of like what you were talking about yesterday, because these guys are leaving a signal, but it's a macroscopic signal, unlike your tiny little microscopic waves. These are macroscopic waves that are left behind by those. And the idea is, could you use them for detection or could you use them for cooperation? The two different coins, the two questions that you were asking yesterday would be applicable at this large scale.
Oh. It'd be very interesting to hear something about which machine learning tools you use to learn the drift and the fusion.
Sure, I'm happy to share that with you.
Okay.
Yes, yes. We're not developing our own algorithms. We're using other people's algorithms, and I'm happy to share that with you. Yes, yes.
Is it because of the symmetry of the confinement? Do you have any intuition?
That's a great question, and I don't have a lot of intuition about. The only thing I know is that those order parameters are by design between 0 and 1, right? So by design, your polarization can be between 0 and 1, your rotational order can be between 0 and 1. what's intriguing to me is that when they transition between one state and the other, they don't go to states of very low polarization and then back. They stay on that outer rim, so to speak. So I don't have a lot of intuition of why they maintain like why they are constrained on this 1D subspace, but it is intriguing, and
So I think we move that question to the break, please. Overrun a tiny bit. So let's thank both speakers this morning.